for them to perform smoothly, like helping each other out, they need to be in the proximity to each other. The thylakoid and the stroma have to be very close to each other, and therefore stroma is found in the surrounding environment of the thylakoid. It contains the enzymes of the Calvin cycle, sugars, and organic acids. Now, if you were asked in the paper in the exam, what kind of enzyme found in the stroma? In the stroma, there will be a lot of enzymes out there, hundreds of them, but at least you guys know one of them. Okay, that is the enzyme that helps the Calvin cycle at the very beginning of the cycle. That is the rubisco. Okay, rubisco. I've shown you here, and rubisco, yeah, somewhere here. There you go, rubisco. Okay, stands for ribulose bisphosphate carboxylase, rubisco. At least you guys know one enzyme found in the stroma, and that's the rubisco. And actually, that is, one out of two enzymes that you guys need to know for photosynthesis. The other being the ATP synthase, the same enzyme helping the process of aerobic respiration. Two enzymes, ATP synthase. Okay, something in common between chloroplast and mitochondria. This is the evidence of the evolution of mitochondria and chloroplast, proving that they have the same common ancestor. Okay, otherwise, how could both organelles, two different organelles, can have the same enzyme and they perform the similar mechanism in producing the ATP, right? Okay, so rubisco and ATP synthase, keep those in mind. Stroma also contains sugars and organic acids. No wonder, because stroma is where the sugars are made. It's where the triose phosphate is made. It is also where the fatty acids are made, amino acids are made. So in a chloroplast, in the stroma chloroplast, we can find lots of organic compounds. Sugars and organic acids are just few of them. Okay, and because it bathes the membranes of the grana, they can exchange things between them. Okay, just to remind you that the Calvin cycle, or the light-independent reaction, requires two things from the light-dependent reaction. They are the reduced NADP and ATP. Those two come from the thylakoid, or from the grana. Now, because they are located in close proximity to each other, the reduced NADP and ATP can go right away towards the stroma from the grana. Then the question is, what does the light-dependent reaction requires? Okay, to happen, they need NADP and they need ADP. Okay, how does the reaction get those chemicals from? From the stroma, from the Calvin cycle. So, as we see here, we have seen interdependence between both reactions. Okay, they're supplying each other with chemicals. The photophosphorylation provides the Calvin cycle with reduced NADP and ATP. The Calvin cycle provides the photophosphorylation with ADP and NADP, like that. And in stroma also, we can find 70S ribosomes. Remember, chloroplasts were actually the ancient prokaryotes, so they have their own ribosomes and set of DNA, a loop of DNA. Here it means that the DNA is circular, lipid droplets and starch grains. Lipid droplets and starch grains. Okay, what is the need for DNA in the chloroplast? Remember, the chloroplasts need to make proteins. What kind of proteins? ATP synthase is one of them. Rubisco is one of them. The photosystem is another example of protein that the chloroplast needs to make. Now, you guys have learned in chapter 6, ACE level, that if you want to produce a protein in the cell, you need a blueprint for it. And the blueprint is provided by the genetic code. The genetic code for making those proteins in the chloroplast are given by the DNA codes stored in the circular DNA of the chloroplast. Okay. However, other chloroplast proteins are coded formed by the DNA in the plant cell nucleus. Again, we are seeing interdependence between the bigger plant cell and the chloroplast. Yes, the chloroplasts provide the plant cell with sugars, with organic compounds, but the bigger plant cell, the host, also provides the chloroplast with some proteins that only the plant cell can produce, but the chloroplast cannot. Right, so those are the functions and the structures of chloroplast. More in details compared to chapter 1 and the previous lesson in IGCS. More details now. Okay, before we proceed further to the next section of limiting factor, I would like to go back here because there are some things that I missed before the holiday. The first is still about the photophosphorylation here. Photophosphorylation is the other name for light-dependent reaction. Now, about this, we have two different pathways. The first one is the non-cyclic photophosphorylation, the one that I taught you before. Okay, it begins with photosystem 2, whereby the water is broken, and then electrons are released, and then the electrons are excited by the photons, and then the electrons are then transferred to power the production of ATP, the way or the same way as mitochondrion produces ATP by, pumping protons, and then the protons flow back by diffusion through the ATP synthase, and ATP is produced. And then the electrons then travel into the photosystem 1, where it is once again excited by the photons from the light, from the sunlight, from the artificial light. Doesn't matter, as long as it has sufficient energy. The electrons then would be caught by a molecule called NADP, and the NADP therefore turns into reduced NADP. Now, that's the non-cyclic photophosphorylation. You guys have learned that. You just have to revise this again, in case you guys forgot. Now, there's another pathway in the photophosphorylation called the cyclic photophosphorylation. In the cyclic photophosphorylation, we only involve one photosystem, which is photosystem 1. We don't involve photosystem 2 here, just photosystem 1. Now, in photosystem 1, as what happens in the non-cyclic photophosphorylation, the photons arrive at the photosystem, and then the accessory pigments would capture the energy from the photon, and then their electrons are excited, and then the energy from their excited electrons are then transferred onto the neighboring pigment molecules, causing a chain reaction until the energy reaches the reaction center. Don't forget the reaction center. That is the primary pigment sitting at the bottom of the photosystem, receiving all the energy gathered by all other pigment molecules. It happens the same way. After that, the electrons are excited, and then the electrons would be carried by various carriers, and the carriers would utilize the energy from the electrons to pump protons, and then the protons would diffuse back through ATP synthase, and then ATP is produced. Now, after the production of ATP, the electrons have exhausted their energy, and then they return to photosystem 1 again. So this is not the same as the non-cyclic photophosphorylation, whereby the electrons originate from photosystem 2, arriving at photosystem 1, and never comes back to photosystem 2. And therefore, photosystem 2 has to constantly replace the lost electrons by breaking down water. In the cyclic photophosphorylation, it doesn't happen because the electron eventually returns to the photosystem. Again, we only involve photosystem 1. And there is no capture of electron by the NADP here. Now, which one happens in the plants? Both happens. However, we need to water the plant since, in the end, the non-cyclic photophosphorylation would exhaust the electrons in the photosystem 2. That's why plants need water. Okay. The Hill reaction is just the explanation about the experiment conducted by scientists in the past, maybe Mr. Hill or Mrs. Hill, I don't know. And it shows the reducing power of the chloroplasts. Okay, the experiment uses a solution to put the solution of DCP-IP and chloroplasts. Okay, a mixture of solution of DCP-IP with chloroplasts. At the beginning, the DCP-IP is blue because it is in the state of being oxidized. Now, when the chloroplast is exposed to light, the chloroplast would reduce the DCP-IP, and then it would turn colorless. We have the DCP-IP in the lab, after years of being not used, it has expired, so we have to buy another one. Okay, but it is easy to achieve. Maybe with your juniors, I'm gonna experiment with this. Okay, so that's the evidence of the reducing power of the chloroplast. When you shine the chloroplast with light, the chloroplast would excite its electrons. The electrons will be released, and some of the electrons from the chloroplast would be reducing the DCP-IP and turning it colorless. Remember, guys, redox reaction. Reduction means gaining electrons. And the DCP-IP gets the electrons from the chloroplast, but only when we expose the chloroplast to light. Because without light, the electrons from the chloroplast will not be released, would not be emitted or excited. Okay, that's it. Okay, coming up next is factors necessary for photosynthesis. This is not different from what we have learned in IGCSE. Nothing different at all. The factors necessary for photosynthesis are many, but we just have to know 3 external factors for photosynthesis: temperature, light intensity, and carbon dioxide concentration. Well, actually, we have more factors needed for photosynthesis. We need chloroplast, we need the opening of stomata, we need the proper thickness of the cell wall, and we need the proper volume of the vacuole, and many more. But we just have to know about 3 external factors for photosynthesis. Those 3, same like IGCS, nothing different. And here in the same section here, we're gonna also learn about the limiting factors. But the limiting factors here are not different at all from what you have learned 2 years ago with me about the limiting factors. For example, this, if you have various light intensity and then you measure the rate of photosynthesis against various light intensities, and then you got these kind of graph here, like this one, you guys have acquired the knowledge and understanding that this part of the graph is where the rate of photosynthesis is limited by the light intensity, and this part of the graph shows the rate of photosynthesis is no longer limited by the light intensity, because the light intensity is no longer limiting since the intensity has been abundant. So there is something else that is limiting the rate of photosynthesis. What could it be? Like I said 2 years ago, you're free to speculate. What factor limits the rate of photosynthesis? Anything other than light intensity. Now, since we have 3 external factors, then that leaves you with 2 other options. Either it is the concentration of carbon dioxide or the temperature. Okay, so you can mention either one of them. The way you answer it on the paper is still the same like before. You say that from light intensity here until here, the light intensity is still limiting, but beyond that, it is no longer limiting, but perhaps CO2 concentration or temperature is limiting. Again, the way you answer it is still the same. Nothing is different. Okay, in experiment 1, you are doing it at 25 degrees Celsius, and at this 0.04% of CO2, the normal concentration of CO2 in the atmosphere. In experiment 2, you are proving that here temperature is the limiting factor because now the plateau of rate of photosynthesis is lower than before. And then here you are proving that CO2 concentration is also a limiting factor. It's also a limiting factor because now by increasing the concentration of CO2 by tenfold, you increase the plateau of rate of photosynthesis. Again, the same as before. If you still remember your lesson in the past, 2 years ago in grade 9, you can apply that again here in A level. And you can still use my notes from 2 years ago for this one. Okay, assuming that you still keep them. Growing plants in protected environments is just like what we learned about the greenhouse or the glass house. By growing them in a greenhouse or glass house, you can control the environment so that you can control the external factors. You can control the temperature, you can control the CO2 concentration, and you can control the light intensity. Anything other than those is beyond your control, like the number of chloroplasts, the number of chlorophyll, and the density of the palisade cells, something beyond your control. You can control only the external factors in the glass houses. Those three. So by doing the modifications, you can make sure or ensure the optimum environment for the optimum growth of plants. You know, in the nature, sometimes plants are limited by the day and night cycle. When the night comes, there's no light, the plants will stop photosynthesizing, and the growth is somehow inhibited or slows down a bit. But if you keep them in a glass house and you can shine them with artificial light, then you can force them to do photosynthesis for 24 hours. It will not make them exhausted. Okay, unlike in humans where you force them to run 24 hours, now plants do not become exhausted if they are forced to do photosynthesis for 24 hours. Instead, you will increase their yield. And also in the glass houses, you can put more carbon dioxide so that they can increase their rate of photosynthesis. And don't worry, when you walk into the greenhouse or the glass house, it won't kill you. That much of carbon dioxide is not enough to kill a person. And the temperature too is kept warm all the time. If the temperature is 25 degrees Celsius as the optimum one, you can maintain that. If the optimum temperature is 30, you can maintain that. So you are in total control of the external factors. Okay? If you guys have no more questions, remember, if you want to ask questions, you can just chat right away. All right, the next section is about a different kind of plants called the C4 plants. The usual plants that we learned for photosynthesis before are called the C3 plants. Here, we're going to learn about the metabolism of C4 plants. The simple difference between C3 and C4 plants are their habitats. C4 plants live in a warmer habitat. The examples are given here, so you can imagine them: the maize or corn, sorghum, and sugarcane. You know, the common crops that Indonesians grow. They are C4 plants. Okay, they are adapted in living in a warmer place, not in colder place. Now, in order for them to cope to such habitat, they evolve a different kind of metabolism, a different kind of physiology. But let me start with what photorespiration is. And then you will see why the name is C4 plants and why the others are called the C3 plants. Now, what is a photorespiration? Photorespiration is a process of reaction or a chemical reaction whereby the rubisco is, instead of fixing CO2 with RUBP, the rubisco fixes oxygen with RUBP here. The reason is a problem with the enzyme rubisco. Yeah, we have learned before since grade 7 perhaps that enzyme is specific. One enzyme can only catalyze one kind of reaction. But don't forget that the reaction can happen due to the very specific shape of the active site. As long as substrates are complementary with the shape of the active site, then reaction can happen. And it turns out that the combination of oxygen and RUBP can fit in to the active site as well as carbon dioxide and RUBP, almost as well as CO2 and RUBP. And therefore, if the Rubisco is exposed to a warmer temperature, to the hotter temperature, you not only expect a faster reaction between RUBP and CO2, not only that, but you also expect the reaction between RUBP and oxygen. That's the downside of Rubisco. Rubisco cannot distinguish sometimes between CO2 and O2. If the temperature is correct, if the temperature is warmer, even oxygen can be catalyzed to react with RUBP by the Rubisco. That's the problem with Rubisco. Okay, now this kind of reaction is called the photorespiration. Yeah, the term can be misleading, but now you have understood what is meant by photorespiration. It's not respiration with the help of light. No such thing, guys. It is just oxygen somehow is fixed onto the RUBP by Rubisco. It happens in the plants, and because it involves oxygen, the term uses the term respiration. But actually, it's not respiration as the breakdown of compound with the help of light. It's not that. Okay, now this kind of photorespiration can waste a lot of RUBP because RUBP does not normally react with oxygen. If RUBP reacts with oxygen, they will be wasted. It will be wasted because that compound, after reacting with oxygen, can no longer be involved in the next step of Calvin cycle. And the number of RUBP available to continue with the Calvin cycle will be reduced. And the Calvin cycle produces organic compounds. The number of organic compounds produced by the Calvin cycle would decrease since some RUBP are wasted. Okay, now photorespiration happens mostly in high temperatures and high light intensity, exactly the condition in tropical countries. Maize, sorghum, and sugarcane, those are the commodities grown in tropical countries, and they can cope with that high light intensity at high temperature by avoiding photorespiration. They can do that. And how? Let's take a look. I may not be able to finish everything today, so let's just get with the introduction of the anatomy and a bit of physiology of C4 plant. Now, what's unique about C4 plant is in the leaves. It's in the leaves. Now, normally we find the mesophyll cells as palisade and as spongy. That's in C3 plants. But in C4 plants, some mesophyll cells are gathering around the bundle sheath cells. You know that these are xylem. The bundle sheath consists of xylem and phloem. The bundle, the vascular bundle, The vascular bundle consists of xylem and phloem, in case you guys forget. Okay, in a C4 plant, the xylem and phloem are surrounded or enveloped by another set of cells called the sheath cells, bundle sheath cells. Now, as if it is not enough, the bundle sheath cells are surrounded more by a ring of mesophyll cells. Surrounded by a ring of mesophyll cells. So these are not palisade nor spongy. They're just ring of mesophyll cells. And just like any other mesophyll cells, they have chloroplasts and they can perform photosynthesis, although a bit different. Okay, the way it does the photosynthesis is a bit different. Now, how does it happen? Let's take a look at figure 13.14 here. We only take one mesophyll cell and one bundle sheath cell. In the mesophyll cell, carbon dioxide is not fixed by REBP, no, but it is fixed by PEP, phosphoenol pyruvate. But you can just call it PEP, phosphoenol pyruvate. Not REBP, but PEP. The fixation is done by PEP. And PEP has 3 carbon atoms. CO2 has 1 carbon atom. After the joining of C3 PEP with CO2, it produces oxaloacetate. It has 4 carbon atoms. Now, this is how the name comes from, C4-1X. It comes from the fact that the first compound after the fixation of carbon dioxide has 4 carbon atoms, as opposed to C3-Plex. Yes, I know that after fixation of carbon dioxide in Calvin cycle, there's a compound with 6 carbon atoms, but it is very unstable, so it's short-lived. Short-lived, so shortly after the formation of that compound, it breaks into GP with 3 carbon atoms each. And therefore, the plants that conduct or carry out this Calvin cycle is called the C3 plants. C3 plants. Not C6 plants, but C3 plants. Once again, because this intermediate compound is very highly unstable. It breaks down the moment it is formed. Okay? That's just to differentiate C3 from C4. And once again, the PEP is the one that fixes the carbon dioxide, not the REBP. And therefore, the name of the enzyme is also different. The name of the enzyme that does this process is called the PEP carboxylase. Easy to remember, isn't it? Okay, it is the enzyme that carboxylate the PEP. Carboxylase is the enzyme that does the carboxylation process, or adding carbon. Adding carbon to who? To PEP. So therefore, the name of the enzyme is PEP carboxylase. This PEP carboxylase is not too sensitive. It's not as sensitive to temperature as the Rubisco. So this PEP carboxylase would only catch carbon dioxide and add it into the PEP, but not the oxygen. Okay, so we produce C4 compound called the oxaloacetate right after the fixation of carbon dioxide. Okay, that will be just the beginning of the whole physiology and biochemistry of C4 photosynthesis, and also the reason why the plants are named as such.